Bismillahirrahmanirrahim. We start with another paper, which is Biology 5090. And this Biology 5090 is going to be the normal O-level biology syllabus that we follow. This is not the IGCSE, this is the other O-level syllabus that we follow, and this will be the November 2020 question paper 1-1, which we are going to discuss is one of the variants. The time of the exam is 1 hour and you have 40 questions, and you have less than a minute for each question. Well, you have a minute, mostly a minute for each question, but then of course you need to revise it, and some of the questions you're going to spend more time. So I always say leave those questions and come back to them later on when you have finished the paper and you've done the questions that you know. Start with question 1. The diagram shows the structure of a cell from a leaf of a green plant. 1, 2, and 3. 1 is the cell wall, 2 is the cell membrane, 3 is the cytoplasm. What are the numbered parts of the cell? Cell wall, cell membrane, cytoplasm. These, of course, would be the chloroplasts, and of course, this you can see is the dark one would be the nucleus. This would be the vacuole, and this will contain the cell sap. So, just a quick revision of what could possibly the labels on this. Then coming on to the next, we come on to question number 2. Now question number 2 is about the diagram shows 3 plant cells labeled P, Q, and R. The arrows show the direction of water movement by osmosis. So water is moving from Q to P, and then from P to R, and from Q to R. Now what is the correct order of water potential in the cells from the highest to the lowest? Now the fact that water is going from Q to P and P to R and Q to R, so of course this would be, say, minus 100. This is how we measured. So this would be minus 90 and this would be minus 80. This is how we measure water potential. So Q would have the highest water because water is moving from Q to P and Q to R. But then water is also moving from P to R. So that means P is more than R and less than Q. So that is why it was Q, P, and R. Question 3. An experiment measured the rate at which plants take up magnesium ions from solution. One plant was given a poison that stops respiration. Now actually what are they examining? Active transport of ions into the root hair cell. And if active transport requires energy, respiration releases energy, so we have to correlate active transport, energy, and the process of respiration which releases energy. Another plant was left as normal. The graph shows these results. So number 1, an experiment measured the rate at which plants take up magnesium from the solution. One plant was given a poison that stops respiration. So no energy, no active transport. Another plant was left as normal. The graph shows the results. Rate of uptake of magnesium ions is on the y-axis. Plant left as normal and plant given poison. So you can see the concentration of magnesium ions in the solution was started to increase from this point here, and this is the point in which the rate of uptake of magnesium ions in the plant given the poison. And then the one which was left as normal, the rate of uptake of magnesium ions is increasing till N, and then it is increasing. So rate of uptake of magnesium ions in a normal plant goes up to N, and then of course, so it's N and P. Now let's look at the questions. The question says, I'm going to rub this off. How are the magnesium ions being absorbed by the plants at points N and P? Now at N, of course, it's active transport, and because the concentration of magnesium ions is in the solution is decreasing as time goes by, because we told you that an experiment measuring the rate at which plants take up magnesium ions from a solution. Magnesium ions from this solution. So we were talking about that solution. So rate of uptake here, the concentration of magnesium ions in the solution is increasing, and it sort of keeps on increasing, then it sort of takes a little, so the magnesium in the solution is more, as it is, because you see the arrow of the concentration of magnesium is in this direction. So how are the magnesium ions being absorbed at N and at P? At N it is active transport and at P it is diffusion, because the plant which was given the poison, there is going to be no respiration, no release of energy. So a time comes when the concentration of magnesium ions is more, then of course it will just going to diffuse into the plant. So this is what we had to understand about this question number 3. For starch digestion occurs in the mouth cavity and in the duodenum, but it stops in the stomach. Why is this? Why does the starch digestion stop in the stomach? Because the pH in the stomach changes the shape of the amylase. Now, where was the amylase coming from? The amylase was coming from the mouth, the salivary amylase. But there is no starch digesting enzyme in the stomach. But why is the salivary amylase not working? Is because the pH in the stomach is so less. It's very highly acidic because of the HCl produced by the cells in the wall of the stomach. So the HCl lowers the pH, and this of course denatures the salivary amylase, which cannot function. Coming on to question 5, which row correctly shows the number of molecules for each substance used and produced during photosynthesis? So 6CO2 plus 6H2O gives you 1C6H12O6 and 6O2. Of course, if you knew the equation, you could do this. Everybody needs to remember the equation for photosynthesis, which is the chemical equation for photosynthesis. Then coming on to question number 6. If you look at question number 6, how do carbon dioxide and water enter a leaf? Carbon dioxide and water enter a leaf. So basically, carbon dioxide enters a leaf through the stomata by diffusion. But how does it enter a leaf? Through the xylem, from the soil. The water is in the soil, and then it enters the root hair cell, and then it's pulled up. Why would it be pulled up? Because as water vapors diffuse out of the leaf, a transpiration pull is created. Just like if you have a glass of water and you put a straw in it, but the water is not going to bubble out of the straw, there has to be some suction force. So if you apply suction force, you take the straw and you suck it, only then the water is going to be drawn up into the straw, which is present in this glass of water. So you have a glass of water and you have a straw in it. So unless you apply some suction force, only that suction force is going to pull up this water. So this water in this glass is going to be pulled up by this suction force, which you are going to create by sucking onto the straw. Then coming on to question number 7, the graph shows the rate of how the rate of photosynthesis varies with light intensity. So light intensity, you can write 1, 2, 3, 4, 5, 6, 7, 8, something like this. 1 bulb, 2 bulbs, 3 bulbs. And the rate of photosynthesis increases. So we can say 10, 20, 30, 40, 50, 60, 70, or 100. So you must always write this, because these papers on which you're doing the MCQ, this paper is going to stay here. Only your answer sheet is going to go to Cambridge to be checked. So the graph shows how the rate of photosynthesis varies with light intensity at 2 different temperatures. So we have one temperature 25 and one we have 15 degrees Celsius. Other variables are kept the same. Now they're asking you some very interesting questions on the limiting factor. In which sections of the graph is light intensity the limiting factor? Light intensity will only be the limiting factor when the graph plateaus. So what we have to light intensity would not be the limiting factor any longer when it plateaus, but light intensity will always be the limiting factor in this part before it plateaus. So let's look at this part. Where is this part? Before it plateaus is this part. Because as you are increasing the light intensity, the rate is also increasing. The rate is also increasing. So that is why the answer would be Q and S. Because once, it could be a little up to here, this point. But once it straightens, then the line straightens, like here, the light becomes straight here, and the line becomes straight here. Now we say light is no longer the limiting factor, because you're increasing light, but the rate is not increasing. So light is no longer limiting in this part of the graph. In P and R, light is no longer the limiting factor. So in which sections of the graph is light intensity limiting? Or if I change this question, if I say light intensity is no longer limiting, then it will be P and R, but here it is Q and S, because they've said in which section of the graph is light intensity the limiting factor. Less light, less photosynthesis. More light, more photosynthesis. Light intensity is limiting. But here we are increasing the light. We have increasing the light for 2 bulbs, 3 bulbs, 4 bulbs, 7 bulbs, 8 bulbs, but still the rate is constant. So light is no longer the limiting factor. So please understand that and be very clear on that point. Going on to question number 8, the diagram shows the human alimentary canal. In which part does protein digestion begin? Naturally, it's the stomach, because in the mouth we only have the salivary amylase. In the stomach we have the pepsin. And the pepsin starts digesting the protein to peptides. So which part does protein digestion begin? You had a piece of chicken and you had a piece of bread. The bread will start to be digested in your mouth when you're chewing it, but not the chicken. The chicken will start being digested in the stomach, and that is the C part of the syllabus. A is the small intestine, D is the large intestine, and B is the mouth cavity, or which is called the buccal cavity. Then coming on to question number 9, blood flows away from the small intestine in the hepatic portal vein. Where does the blood go next? And what may happen there? So we all know this would be wrong. The kidney is wrong, but it has to be either C or D. The liver, where alcohol is broken down. The liver, where bile is added to the blood. You know, bile is added never to the blood. Bile is added to the small intestine. Bile is produced by the pancreas, by the liver, stored in the gallbladder, and then it is added to the food in the duodenum. So liver, where bile is added to the blood, I mean that would be sort of a child who didn't know this, is not added to the blood, is never added to the blood. Then coming on to question number 10, the symptoms of a disease include weakness, fatigue, aching and swollen joints, and swollen and soft gums. And that is which food is used to treat this disease. Oranges as a source of vitamin E. Basically, they are telling you all these signs and symptoms of scurvy. Weakness, fatigue, aching, swollen joints, and swollen and soft gums. So, what do we need? Scurvy is a disease of deficiency of vitamin C, and this is what we had to figure out. Then coming on to question number 11, which diagram shows the position of phloem tissue in a herbaceous dicot root? Now this of course we know is the stem. Why? Because stem has the vascular bundles out on the periphery. The root is the one which has it in the center, shows the position of phloem tissue. We know the X in the xylem. This is X is the xylem, and then D is the phloem, and this is what we have to know. This is something factual that we need to understand and remember. Then coming on to question number 12, the diagram represents part of a leaf, and we have one, and the one is coming here somewhere, and this is of course the palisade mesophyll, then 3, and then we have 4, and then we have 2. It says which arrow shows translocation and transpiration? Very interesting, very good question. You have to know that the xylem has got this lignin, so this is the xylem, and in the leaf it is the upper which is xylem. That's the mnemonic for that. And this is the phloem, because you can see the sieve plates in it. This is 3 would be the phloem, the sieve plates in it. So that is why translocation would be 3. And then it says transpiration. Why would transpiration? It says which arrow shows translocation and transpiration. So in transpiration, we are seeing this water molecule which must have been present inside this cell. Then it has evaporated, then it's formed a water vapor, and then the water vapors diffuse out through the stomata. And in 2, what is happening is actually this is carbon dioxide moving in and photosynthesis is taking in. So this would be the movement of carbon dioxide. Transpiration is the evaporation of water from the spongy mesophyll cells. First, it has to move out by osmosis, then it has to evaporate from the surface of the mesophyll cells, spongy mesophyll cells, and then the water vapors have to diffuse out. So that is why the answer would be 3 and 4, which is C. Then coming on to the next question. which is question number 13. In the process of blood clotting, D1 help to convert 2 to 3, which forms a clot. Which word is complete? So what do we write here in this space? What do we write here in this space? And what do we write here in this space? So in the process of blood clotting, the platelets help to convert fibrinogen to fibrin, and that is what is the important thing. Fibrinogen to fibrin, which forms a clot, and of course that was only we were going to confuse this with B and D. So it's not red blood cells, it's platelets which help to convert. Platelets release an enzyme, thrombokinase. Thrombokinase converts prothrombin to thrombin, and the thrombin converts fibrinogen to fibrin, and that is something which we have to learn, and we have to revise that if you don't know that as yet. Then coming on to question number 14, the diagram shows a section of a capillary. Which arrow represents tissue fluid formation? And they've given you some clues. They've said this is the arterial end and this is the venous end. You know, the arterial end, the pressure is very high. The blood is coming from an artery. The pressure is high, very high blood pressure, hydrostatic pressure. And this, of course, causes tissue fluid to be formed. And this results in the plasma, which was inside. Once it was inside the blood vessel, that same fluid is called plasma. And as soon as that fluid comes out, it is called tissue fluid. So what we had to understand is on the arterial side, this is the arterial side, the pressure is high. So the answer to that had to be A. Then coming on to question number 15, which row shows possible causes of coronary heart disease? And the options are diets high in saturated fats, diet low in salt, and regular exercise. So the answer was only the tick was in the high in saturated fats. Of course, if a person does regular exercise, that helps, and people who take less salt in their diet will also help and will not have a greater chance of coronary heart disease. Then question 16, what are the products of anaerobic respiration in yeast? And that's alcohol, carbon dioxide, and a low energy yield. So yeast, you know, always produces alcohol and carbon dioxide, because remember when we're doing bread making and the dough rises, that's because of the carbon dioxide, and it releases less energy. All forms of anaerobic respiration release less energy. Then question 17, the diagram shows apparatus used to investigate respiration. Respiration is the release of energy, and what is going to be happen is glucose plus oxygen gives you carbon dioxide plus water plus energy. So we've got a diagram to show investigation. We've got temperature, we've got germinating seeds, we've got a wire mesh, we've got a sodium hydroxide to absorb the carbon dioxide. We have a colored oil drop, and it says which changes will be seen, and what is the explanation. So which changes will be seen and what will be the explanation? The oil drop will move towards the left, and the explanation is oxygen is used up by the peas. Because you see, the oxygen in the air is being used up by the peas, but the carbon dioxide which they are producing is going into this chemical which is absorbing the carbon dioxide. They could have also shown you a diagram in which they could have shown you an increase in temperature in the thermometer, and that would have shown you, of course, another explanation. Oil drop moves to the left, but then of course we won't use an oil drop move to the left and the temperature rises. Then of course the explanation would be oxygen used up and energy released. So this is how we had to be looking at this question, that the oil drop moves to the left because this air inside this contains oxygen, and that oxygen is used up, and when that is used up, a pressure difference is created. This of course forces the oil drop, and the answer to this is because of the pressure difference, this oil drop moves to the left. So please understand this question, because I can easily think of them changing this question in your final exam in the end of July. Then coming to question number 18, the diagram shows the rib cage and some of the muscles involved in breathing as seen from the side. What have we got labeled here? Vertebral column. So this is the back. And then we have the sternum, which is the breastbone, which is in the front. And then we've got the intercostal muscles, which join these ribs, external and internal. And then we have the diaphragm, and then we have the ribs again. They've shown you the diagram of that. They've labeled all this. What happens when the intercostal muscles shown in the diagram contract? Now, that's a very difficult question, because if you don't know the direction of the intercostal muscles, you see they are like this. This is what you have to understand. These are the internal intercostal muscles, and when they contract, they are going to pull the ribs down. So please remember, when you inhale, we say external intercostal muscles contract, pull the rib cage upwards and outwards. But then they've said which ones contract, because they're asking you these, they've shown you these, and these are the internal intercostal muscles, and the ribs are going to move down. So this is what you have. If they're going to move down, this is going to result in decrease in the volume and increase in pressure, and the air is going to be exhaled out. So please go back, put in the Google search the external intercostal muscles and see their direction. You see the external intercostal muscles are in this direction. They're towards the sternum. And these are, of course, to the other side, because when they are going to contract, they're going to move the ribs down. And this is what you need to understand about this. This was a difficult question, and this is the one which is going to decide what grade you get. Then, of course, comes to question number 19. During dialysis, substances such as glucose, mineral salts, urea, and water move between the patient's blood and the dialysis fluid. Which processes are involved in these movements? So the answer was, there's no active transport, there's only diffusion, because the blood is on one side, so the blood is circulating inside these vessels, and outside this we have the tissue fluid. So the glucose does not move in or out, it's move between. So glucose, there's no actually net movement, mineral salt, but urea moves out of the blood into the dialysis fluid. And then the dialysis fluid is changed actually all the time. And this, of course, is a movement of diffusion. It isn't a movement of any active transport, because the blood is out of your body, out of the patient's body, into the machine. So how can there be any active transport there? So that is why there is no active transport, and yes, it is diffusion. So this was the line which was correct, and you had to understand this. Now we come to question number 20. Which organs in humans are involved in maintaining body temperature? Brain and the skin. Yes, both. Both are involved. Why? Because the brain has got the centers which control your body temperature, and the skin has the blood vessels which are either going to vasoconstrict, the arterioles vasoconstrict or vasodilate. Please remember, it's the arteries and the arterioles, not the capillaries. Capillaries cannot contract and relax. They cannot alter their diameter because they do not have any muscles in their walls. There are no muscles, and these are smooth muscles in the wall of the arteries and the arterioles, which of course contract and relax to alter their volume. And then we come on to question number 21. The diagram shows a section through the brain. Which part is the hypothalamus? Now that's labeling. Now you must look at different diagrams and you have to just label it. It's the area just above the pituitary gland, and D is the pituitary gland, and B is the cerebellum, and C is the medulla. So you have to understand how the labeling is. You need to just put in the Google search diagrams of the structure of the brain, and you can see a whole lot of diagrams and see different sort of versions of it and see all the labels on it. Next, coming on to question number 22. Now 22 is a diagram of the eye. The diagram shows a section through the eye, and this is up and down. When the person looks down and focus on their mobile phone, what is the state of the structures P and R? Now, what are P and R? R are the suspensory ligaments, and P is the ciliary muscle which is inside the ciliary body. So P is contracted and R are loose. Why? Because it slackens the suspensory ligaments. When you're looking at near, the lens becomes more convex. When the lens becomes more convex is because the ciliary muscles contract. And this, of course, makes a smaller circle, so the suspensory ligaments loosen up. Please look at the video which I have on this chapter and then see and revise that chapter. And I'm sure you can understand that why is it contracted and why are they loose. It's either we say the words taut and slack, but here they've used the word loose and tight, so they've sort of become a little loosened, or they're taut or they're tight and pulled tight. So this was question 22. Then we come on to question number 23. What is a sign of diabetes? Sign, is something which we can check and see. Symptom is what you feel. Like headache is a symptom, and I cannot tell if you say you have a headache. Well, I have no way to check. I don't have a thermometer or something which I can check that you have a headache or not. Sign is like if you say I have fever. Well, I've got a thermometer. I'm going to ask you to just keep it in your mouth or your axilla, and of course, I'm going to check your temperature and know whether you've got fever or not. So B, the sign of diabetes is glucose in the urine. Please remember, normal people do not have any glucose in the urine. And if you have glucose in the urine, glucose in the blood is present all the time. You always have glucose in the blood. Of course, if you have higher levels of glucose in the blood, that would be a sign of diabetes, which is, of course, your highest goes up to 200. And but if you have it like 400 or 300, then you have diabetes. So glucose in the blood would is something which is glucose, not just glucose in the blood, but higher levels of glucose in the blood. Normal people also have glucose in the blood. We all have glucose in the blood. If you don't have any glucose in the blood, you will probably die. Okay, so that finishes this up to here. We will continue this in the next video. And thank you for now, and all the very best.